Good morning, everybody, and welcome. Thank you all for coming here, and thank you, Dementia Research Network Ireland, and Karen and Calvin, for all your work that you put into these seminars. They're really very interesting and well planned as well, given our circumstances. So, my talk today is on neural engineering for rehabilitation within neurodegenerative disease. So I'll discuss about certain key findings in the areas that I've been involved in. So do let me know if you have any questions at the end. So obviously neurodegenerative disease is very complex. In fact, Dr. Barry Boland was discussing previously in this seminar series how 600,000 people within Ireland have a family member affected by neurodegenerative disease. So it is a complex problem and reaching a lot of our population. It obviously has aging is a major risk factor within it. In most neurodegenerative diseases, including Alzheimer's disease and Parkinson's disease. So this is obviously becoming more of an issue as our population ages, which is a wonderful thing, but it causes so our health system some problems. The two areas I'm going to be focusing on today are the motor and the cognitive function within neurodegenerative disease, and how this is impaired, and how we can quantify this so that we can allow a screening perhaps to occur earlier on in this disease in order to perhaps be able to clinically do something before some of the function has gone. So our research questions that we have really are within this area is how does the brain control movement, and the pathophysiology of neurodegenerative disease? Can we understand control of movement, and what are the associations between cognitive and motor impairments? Is this a direct link? Or is this an indirect link due to a function within the system? So exactly how does this occur? What is the what strategies do people put in place themselves? There's been a lot of research looking into categorizing this in neurodegeneration further. So looking at motor phenotyping, looking at the different areas of gait and the different parameters within gait, and trying to assess if different areas could perhaps be linked to different cognitive areas. And so this relationship is very interesting, because if we can find a link between this, then possibly is this a bidirectional link, and can we have some improvement within this? Obviously looking at intervention, so looking at the aging brain is very important, the healthy, normal aging brain. And it is really very important that we look and that we find a point that this normal aging brain turns and changes from something that is degenerating. And if we can learn more about this, then perhaps we can learn more about this rejuvenation of this, or rehabilitation might be perhaps possible. So my main area of research looking at gait, so it's very important to look at gait in normal aging, because then we might have a baseline. So we do know that there's a general slowing of processes with normal aging. Our gait slows down, or the amount of time we put our feet on the ground increases, so our support times increase, and our the length of our strides reduce, and our the width of our stance increases. And this is obviously to maintain stability, but gait speed and other gait factors have been found to be predictors of different aspects. So a big study in 2011 found that gait speed was a predictor of survival. And then more recently, the same author, Studensky, looked at clinical meaningful outcomes there. So reassessing this 8 years later and still finding that this is a very interesting thing to be bringing into the clinic, and she calls it bradypedia. So in order to, are these changes within gait to maintain stability, or could they be a marker of something more important? So there's also links between cognitive function and gait. So certain studies have found that you need to look at the two together in order to actually really be able to parse apart this relationship, and look at the two together in order to find risks and perhaps allow people to be screened more easily and at an earlier point. So how do we design studies or interventions to capture motor and cognitive function? So there are several ways, and myself as an engineer, obviously, I'm always looking at objective measures in order to quantify this. So we can look at gait analysis. So within this, depending on what you're looking at, you want to examine different aspects of gait. So temporal aspects of gait have been found to be of more interest to neurological issues in Parkinson's disease, and then gait speed can be seen as more general health. So depending on what you're looking for, you can design your experiment to be more simple or more complex. So do you want to have just a quick walk, a timed walk, or do you want something much more elaborate that looks at variability and that looks at a longer walk, or a free living environment perhaps? It might be something that you might need to look at. There's also sway, so if you're more interested in falls or balance, you can then look at sway or center of mass. There's also a speech analysis that is very interesting to look at as well. Similar to gait, we're looking at the differences within the signals. And so you would get someone to recite a passage and look at the pauses and the different characteristics of speech. EEG is also another means of that we have used a lot in our studies. And I suppose the benefits of looking at EEG is that it is very, very has very, very good temporal resolution. And for looking at neurological disorders, this is very important. So usually the systems are, you have an awful lot of electrodes on the scalp, and in previous times, these were very, very cumbersome and very heavy, and suffered a lot from movement artifacts. But more recently, there's more a big research push into making this more mobile, and analysis and methods to reduce this movement artifact. So this is really very important because we can assess cognition at the same time as moving. So in order to do this, and in order, so someone may come into the clinic and they may say that they have a certain issue, but it's really very difficult in order to assess that and assess what they're talking about. Perhaps they're walking around their house with a glass of water and they have an issue there, and they're explaining this to their clinician, but so it's very, very difficult to bring that into the clinic. So part of what I'm very interested in is looking at these ecologically valid environments, looking at paradigms that we can bring into the clinic in order to assess participants and patients, to try and assess them in similar environments that they would have at home. So part of this is designing an experiment, designing a task that they may stress the person into doing something that they may have done at home. So for instance, with this experiment, we looked at Parkinson's disease and people with Parkinson's disease that froze. So we had them walking around a maze and pressing a button, and we looked at their EEG and their step. Now, previously, as I mentioned, when you look at complex moods and neuropsychological measurement, a lot of the studies had people very, very still on a treadmill or on a bicycle, and this allowed you to look at certain aspects, but not full movement. So the systems have got more complex, and you can add obviously different movement analysis systems. So at the top here, you see different cameras in a Vicon system with EEG, with a screen, with a treadmill, and then similarly that was in 2010. So more complex systems have come in more recently. And you can have treadmills that move sideways and forward, and you have different systems that perhaps look at how your brain looks at acceleration and moving backwards. But so all these systems, we can now look at EEG while walking, and we can do it, and the data is relatively clean. So we will get a participant to do a test, and they do some within the test, and we have stimuli coming up on the screen. For instance, in the virtual reality study that I was involved in, we had a Stroop test coming up. So a standard stimuli would come up 80% of the time, and a target would come up 20% of the time. So during that 20% of the time, the EEG would show a peak at that time, and you look at the difference between the standard and the target. So this data has been shown more recently to be easier to attain, and this is data from a study I was involved in looking at EEG during movement in Parkinson's disease. There's other systems as well. EEG is the gold standard, but there's other systems such as FNIRS, and these can be useful when you're looking at different areas of the brain, and perhaps if you want something that's maybe a little bit more less expensive, and if you're particularly looking at maybe frontal lobes and you don't want a whole brain map. So these have also been found in recent studies to be a certain amount of accuracy in collecting them in disease populations such as Parkinson's disease. So previously, a lot of EEG, because of the complexities, because the movement artifact, a lot of the studies were done in healthy individuals, but now more so they're being done in diseased populations. So it's very important to be able to obviously have an ecological environment, allow the participant to look at be able to view the participant in an environment that they perhaps would have at home or while walking down the street. So virtual reality has become very important in examining this within participants. It allows safety because you can have the person thinking that they're moving, but allow some sort of a safety for falling in those populations. And you can take neurophysiological data, which is representative of what perhaps they might feel at home. So key studies and findings that I've been involved in, I'm going to go through this quite quickly. So obviously it's very, very important to have a baseline for all this work, and the Irish longitudinal study on aging is of enormous benefit to research in this area. When you're looking at disease populations, obviously looking at this data, it can be very complex, and in order to have a population and a data set that you can view, and and that it's from a population of a similar age with similar comorbidities, is really very important. So initially looking at the TILDA data set, obviously a lot of people here might know of this, and mainly I'll be looking at the gait assessment and the neuropsychological assessment within the TILDA data set. So they have gait assessments within TILDA. They use an electronic mat. It's 4.88 meters long. The person takes 2 passes or goes up and down this mat twice. It has they have a single task, so the person just goes up and down, and then they bring in these dual tasks, so the person walks up and down reciting alternate letters of the alphabet, so A, C, E, and so on. And then they had in their first wave that person carrying a glass of water, but subsequently that has become a faster walk. This was changed in order to stress the participants a little bit more, because they are healthy participants. So perhaps carrying a glass of water might be a good study to have in a very in a population perhaps that wasn't as functional. And the neuropsychological assessments also in TILDA, there's a long battery of assessments. So this is they're using pen and paper tests such as the MMSE, or the Mini Mental State Examination, and the Montreal Cognitive Assessment. So for people that don't know these studies and these assessments, the participant is given this assessment, and they are with the nurse or the clinician. They are asked to perform certain tasks or to name certain things, and they're given a score for this. And that assesses the cognitive function, and also certain domains of their cognitive function. So the whole test adds up to the global cognition and the individual different aspects within it. We can look at their cognitive domains, such as visual, spatial, or executive function, memory, attention, and so on. So looking at this within TILDA, we found that gait speed in all tasks was associated with certain neuropsychological tests and certain cognitive domains. So processing speed, short-term memory, and sustained attention was associated with gait speed in all gait tasks, and particularly executive function was associated with dual tasks, which was very interesting to find. This was cross-sectional data from TILDA, and it was really great that we found that actually the dual task was the dual task had an association with executive function, and therefore was doing what it was supposed to, which was really pushing the participants and trying to find out how well they were doing with their executive function. And could then, we thought from this, could then a dual task be brought in clinically as a more objective and quantitative examination of cognitive tasks? And if this was the case, could we bring in other dual tasks that might allow the person to be screened for different other cognitive domains, such as memory or sustained attention? However, longitudinally looking at this, they didn't find a difference in decline in cognitive function and timed up and go or the gait task. It'll be interesting to see later on and further along in tilde and the other waves, is this still the case? Or and why is that? Is the population healthy, and is it only in diseased populations that we find this link between gait and cognitive function? So speech analysis has also been done by Dr. Céline Deleuze as part of MISA, the Mercer Institute for Successful Aging. Céline is now in Tilda, but she did very interesting work on speech and looking at, similar to gait, looking at transitions from healthy aging to mild cognitive impairment. So looking at people saying different sentences and seeing the different pauses and annotations in their speech, and these speech characteristics, are they indicators of that cognitive impairment? So Céline found, yes, they were an indicator of mild cognitive impairment, in particular certain aspects of speech were very sensitive to this. So other studies having all these findings, really we wanted to see if this was the case, if there was a link between gait and cognitive function, if this was this just due to the fact that both aspects are indirectly linked, and that they or do they are they directly related, and do they are they controlled by similar neurological processes? So in order to see this, we wanted to bring it into the clinic and have an intervention in a disease population that had very poor function because of gait problems. So we, within the VERVE study, we examined people who had freezing of gait in Parkinson's disease, and we examined a dual motor cognitive virtual reality training within this population. So freezing of gait, for people that don't know it, it's a gait disturbance where a person's feet is glued to the ground. Sometimes they're conscious of this and sometimes not, and it is more apparent in the late stages of Parkinson's disease. So there may be an indication for levodopa or certain medications having complications here, and it's really very important to look at because it affects 60% of patients with Parkinson's disease, and people get freezing of gait when they initiate gait in narrow passages, when they go through doorways, when they turn around, and during dual tasking. And this is really very important because it causes falls, and it can cause situations whereby also perhaps someone's crossing the road and they stop and they freeze, and it really impairs quality of life. However, it's poorly understood. There's no effective treatment, and it is very difficult to study because a person can freeze an awful lot and then go into a clinic and not do it so much. And there are certain questionnaires for it, but it's really quite hard to characterize. And it is associated with executive dysfunction and attention as well. So I was involved in a study looking at an intervention on this, whereby a person would step on a balance board, which is called stepping in place, and they had EEG taken at the same time while they were going through a maze. So they stepped in place in order to go through the maze. When they stopped stepping, they'd stop in the maze, and they were also doing a Stroop task as well. So it was a definite dual task. It was a multitasking. And we looked at, obviously they were assessed before and after the intervention, and then they did 8 20-minute interventions. So this was done in 20 participants with Parkinson's disease, 13 of which had freezing of gait. And we looked at different pre to post intervention measures. So stepping in place, cognitive function based on behavioral measures, the reaction time pre and post, and obviously the EEG, and then other clinical measures such as the modified timed up and go and the freezing of gait questionnaire, and also disease rating scale UPDRS, and other neuropsychological measures too. So the main findings were that the training did impact dual task performance in freedom of gait, which was a brilliant finding. And even though it was a very small cohort, the participants really came out with being very the main thing I got from this is the participants really liked being involved in the intervention, and they it was given that it was a small number, hard to parse apart the exactly what was I suppose improving, but there was specific improvements in cognitive and motor parameters. The stepping time and the rhythmicity was a definite improvement in those, and in the dual task effect for those with freezing. But the non-freezers also had certain improvements, but definitely with the freezing of gait, they had better improvements. And the improvements manifested in clinical measures as well. So subjectively reporting improvements in freezing of gait. And so really it would be great to do a study, a much bigger study, because these subjective improvements were found by all the freezers, and it would be nice to continue this more. But it was really showed us that virtual reality interventions are could be used in the home, and they can substantially improve quality of life for patients who experience freezing. Also, we found that it was nice to see the response times that looking at the experimental setup, that actually the stepping in place reduced the response time. So the clicks and the timing of the clicks were reduced for those with freezing of gait. So it's very interesting to see there what is exactly happening there, and this did not happen for the controls and those without freezing. And in addition, looking at the EEG data, that there was the stepping in place did cause differences in EEG to those that compared to sitting. So there were larger peaks, there were earlier onsets while walking, which will also indicate more recruitment of resources for those tasks, which is a good indicator that the dual task and the stepping in place is really pushing the participants. So I suppose really just to discuss how are we going to use this data, could we possibly use it in the clinic, and also in a community setting, given that there's a large aging population, that neurodegenerative disease is such an enormous issue. Can we use it as a screening tool, an early detection for mild cognitive impairment and Alzheimer's disease? So looking at research in this area internationally, Joe Verghese in the Einstein Institute of Aging in the Bronx in New York is looking at mature cognitive risk, and shown that people with this, so this is people with a slow gait as well as a cognitive coming to the clinic with a cognitive issue, and looking at this and bringing gait into the clinic, into the assessment for mild cognitive impairment, shows that those with this cognitive risk are associated with a risk of dementia, and much more than slow gait alone or a cognitive complaint alone. And we looked at this. So another study by Verghese also showed that globally, this MCR has a prevalence of around 9% based on different studies. And Fiacre Maguire looked at this within the Tilda data set, and we found this to be around 2.6% within Tilda, the prevalence of MCR. So it's a lot lower within the Irish population. But we also found that this MCR has does have associations between with cognitive, the neuropsychological assessments within Tilda, which is encouraging, and we'd like to look at this MCR in further waves of Tilda. So bringing this into other populations who have cognitive impairment, so exploring novel biomarkers of brain disease and diabetes. This is a study in Tallaght University Hospital called NBIND, with Adam Dyer and Sean Kennelly, and looking at type 2 diabetes and development of cognitive impairment within type 2 diabetes. People with type 2 diabetes have a much higher risk of mild cognitive impairment and dementia. And how can we perhaps screen these people earlier on? So we've this study has a gait aspect to it, and we're in the process of looking at that data. Adam Dyer has a paper in review looking at early cognitive decrements in the diabetes population, and we're hoping also to look at the gait data within this, and perhaps from that maybe bring in a gait within the clinic. How could we perhaps how could an early career researcher perhaps commercialize this for the community, or bring it within the clinic, and have something that is very objective and quantitative. So I suppose looking at I've been involved in several very interesting studies, but looking, I suppose, my career to date. So I started in engineering in Trinity, and then I did a master's in bioengineering, and I moved from that into industry, into regulatory and compliance as a validation engineer in Abbott Laboratories first, and then into a consultancy role in another validation role too. And from there, I moved to UCD, commercializing college spinouts. And it was at that point, I really always wanted to be involved in research. So at that point, I started my PhD, and as a mature student after working for several years in industry. And from there, did two postdocs, and now I have my role in the Technology University Dublin in Bolton Street in mechanical engineering. So I suppose I've come from industry into commercialization and in research, but really all the skills that I've used and the skills that I've gained within my PhD and within my engineering degree, I've used throughout all those roles. And I've kind of dipped in and out of education, and I have gone to industry, as I said, and come back out, and perhaps in future may go in again. And I suppose what it is the continuous training is really very important. And as a researcher, you have really important skills, and really I think researchers need to be more confident about what they bring to the table. And the future plans for my lab are to get more funding and push this research on further. So if anyone's interested in that, please email me. But I suppose if you what I would like you to take away from this is to be confident in your skill set, and that you may have different settings, but the core activity of designing experiments, collecting and examining resultant data, that can be used in an awful lot of roles. So, I mean, in academia, in the community, and in industry, and development and generation of innovation in no matter what you're doing. So that would be very important. Data science is a massive big role now that a lot of different companies are looking for, and researchers would definitely be able, would excel at those roles. And indeed, further research is, I would say that it is definitely worth pushing at that if you have a genuine interest in that, and you will use the skills, let's say, if you wanted to go and do a PhD, you would definitely use those skills throughout your career. So if anyone has any questions and they don't want to bring it up now, I mean, you can contact me, isabelle.killane@dublin.ie. And just I'd like to acknowledge all the different researchers that were involved, and all the support from all these studies, and that thanks to everybody for allowing me to talk about their research within this talk. So, Carol, if I could.
Yeah, I'm back. Yeah, great.
Right, just a little bit over, sorry.
No, that's okay. It was very interesting. I hope that was okay for the audience as well. But it was just so interesting to hear about, well, certainly all the research that I find that really interesting. You know, the impact of technology and coming into the lab, and then, you know, the development of interventions, and that, as you said as well, the potential for commercialization as well, which is an interesting point. And I think really nice for people to hear as well about the skill set that, you know, researchers have, that they're very transferable into industry. And, you know, you've had an interesting career path in going to industry and back to academia, and, you know, that can be done, which is really nice to see. So we have a question here. How did you find using virtual reality in a research setting? Was it well tolerated by participants?
I would say yes. I mean, yeah, I think in a few studies that I've been involved in, the researcher's idea of what someone tolerates or has an acceptance of is quite different to what the participant accepts. So I think definitely your protocol is very, very important, in that you say the same in order for your outcomes to be, you be able to stand by your results. You need to say the same thing to each person, so your protocol needs to be the same. You need to have definitely have tested it with young, healthy participants, and maybe even control older participants to assess that. But yes, I think it was. And I mean, there were. I mean, there's a lot of research that was involved in that project that I didn't discuss about immersive environments. And there were other studies within that VERV FP7 European Commission project in France, who had very complex virtual environments. So one thing I would say is that the paradigm perhaps has to be designed very well in order for you to be able to see the outcomes, and simplification of the paradigm is paramount in order for you to understand what is coming out of the virtual environment. I don't know if that answers the question.
Okay, yeah, great. And there's probably a certain amount of putting research participants at ease, I guess, you know, if they're coming into a strange environment with new technology. You know, there's probably those softer skills, I suppose, of, you know, making sure that people are comfortable and given time to adapt and all that, you know. So
yeah, I mean, there is There's obviously the safety issue, which you have to, we had a frame, and one person was standing beside the participant. So that, I mean, that is important. At the same time, you do want to evoke, we wanted to evoke a freezing episode, which we did. So that's very important too, and I think the fact that we did that, and the fact that that couldn't be done clinically, is a big benefit of the virtual environment that we had, and that virtual environment. So yeah, I think it was tolerated by participants. We had other experimental setups where they actually participants went into a much bigger setup, and that didn't work as well. The stepping in place for that disease population was more well tolerated than perhaps the Vicon, where they might have to wear, wetsuit type suits and have motion capture, devices on them. So I thought the steps in place.
Okay, great. And just a comment there, thank you for an excellent talk from somebody, so thank you. So I think that's all the questions. So just to say, again, another thank you coming in there. So yeah, thank you for allowed mine. Thank you. Thanks very much, Isabelle, for your time. That was really interesting. And just to let everyone know, so the next talk then is this day next week, same time, and it's a talk by the HRB in relation to funding opportunities for early and mid-career researchers. And in terms of feedback, I'll get feedback at the end of all 6 sessions from participants. So thanks everybody for joining in, and thanks again, Isabelle, and hopefully see you next week. Okay.
That's great.
Take care. Bye.